All right, so let me start by thanking Susanna for the introduction and to thank Nigel for the invitation to be here. I'm actually going to start, well, I guess I started with the thanks. The second part is a disclaimer, which is that this is a talk I have not given before. There are some parts of it I've given before. There's a big chunk of data that I haven't presented. That's our JGI funded research. And so I think I have the timing down, but I'll ask for your indulgence if I go a little bit faster or a little bit slower, depending on how much time is left. I'll also point out that you can see right on the first slide I have that the presenter declares competing financial interests. I'm going to talk about a pathway to make a compound called glucaric acid, and I am a co-founder of a company that's licensed that technology, and so I like to be very upfront about that. I also usually say the interests aren't that competitive. So I still have hopes, but in the interest of full disclosure, I have commercial activities as well as academic activities for this work that's being done. So as Susanna said, what we're interested in my lab is using biology to do chemistry. And I don't think I have to explain why we're interested in that for this audience. That's very nice. But it's helpful, I think, for me to think about, as Susanna mentioned, my undergraduate degree was in chemical engineering at MIT. And when I was an undergraduate, my undergraduate research was in bioprocess engineering. And this was sort of our view of a cell. The cell was just the thing. And what we were largely doing in chemical engineering was designing processes around that thing and trying to design things like media formulations, feeding strategies, oxygen strategies, to be able to improve the productivity of that thing. Well, by the time I got to graduate school, this became the thing, where it was recognized that the cell, from the perspective of an engineer, could be considered to be a collection of chemical reactions. And so you've all seen these slides before. This is from KEGG, where each of the data points here represents a metabolite, and the lines between them represent chemical reactions that are mediated by enzymes that give the cell the collection of both small and macromolecules that allow it to function and also to have certain physical forms. So the challenge we're always faced with is that we have this complexity that we're dealing with, and we want to take some simple starting molecule like glucose and figure out how to navigate that network through a specified set of reactions to get a specific target compound of interest, keeping in mind that there are many byproducts that we don't necessarily want to have to deal with. And in graduate school, when I was a graduate student, I had never heard the phrase synthetic biology before. We were all metabolic engineers. And a hallmark of metabolic engineering is that when you do identify byproducts like this, then you figure out the enzyme that's encoding, the gene encoding the enzyme that's making that unwanted product. You knock out that gene, you eliminate the byproducts. But we also have to deal with the fact that we are frequently competing against growth, that is, being able to use one of our intermediates in the pathway for what the cell really wants to do, which is to take that carbohydrate and make more of itself. So we don't have the option, if growth is our competition, to simply knock that out. Okay, so with this background, our fundamental challenge here is navigating this complex network to maximize productivity of whatever the specific compound is that we're interested in, while minimizing both the unwanted byproducts and what I'm describing as unnecessary growth. So we recognize that growth is essential for giving us sufficient levels of those enzymes to have the catalytic activities that we're interested in, but we want to be able to have a balance between those. So we've worked on the pathway to make glucaric acid for a number of years. It's the first project that I launched in my lab. Just to give you some highlights, many of you may have seen this, especially given your affiliation with the JGI. It was identified in the WRPE 2004 DOE report on top value-added chemicals from biomass as one of those top 12 to 14 molecules. It is a natural product. It's found in fruits and vegetables and a number of mammals, but there's no known microbial pathway for it. So that's what we really set out to do, which was to figure out, if I went back to my training as an undergraduate chemical engineer and imagined putting this cell into a stainless steel reactor, then how could we design a microbial pathway to be able to make the compound of interest? I'll point out that as part of actually the commercial interest that I mentioned before, I've learned that there's quite a bit of use of this molecule in pharmaceutical synthesis. As well, with work in the company, we've done some collaborations where we have found that it improves the properties of various polymeric materials, and it's also useful as deicers or phosphate-free detergents. And there is a chemical synthesis from glucose that relies on a nitric acid catalyzed oxidation. So with that as background, we set out to come up with a simple pathway to allow us to produce this compound in a microbe. We chose E. coli as our host organism of interest. And I mentioned the fact that it is a natural pathway, but the elucidated pathway, which was actually from citrus, consists of about 10 or 12 reaction steps. It interfaces with nucleotide metabolism, so there are UDPs that are used to activate the various intermediates and so on and so forth. So I used to always say it's a fine pathway for its purposes, but not a great one if you're trying to start your lab and ask a graduate student to do this, and you actually want them to stick around long enough for it to happen. So instead, we relied on the fact that because it is a natural product, we could find references of the conversion of this molecule in metabolism databases. And in particular, we found reference to an enzyme called urinate dehydrogenase in a soil bacterium in Pseudomonas syringae that could give us glucaric acid from glucuronic acid. And it turns out this organism can grow on either of these molecules. And we ended up being the first to elucidate the gene, or to identify the gene associated with that UDH activity. And we actually relied on the fact that the cell can grow, E. coli can actually grow on either of these as well, but through two totally distinct pathways. And so we could actually knock out the ability to grow on glucuronic acid, but supply that as the sole carbon source and then use that as a growth-based screen to identify gene fragments encoding this urinate dehydrogenase activity that would allow the cell to grow on glucaric acid. So that was great. We found that gene. As far as Pseudomonas is concerned, that was the end of the story in terms of a pathway. It seems like glucuronic acid is primarily used as a carbon source and not as an end product metabolite. But if we continue to look, we found that glucuronic acid could be produced from myoinositol, and that's actually an enzyme found in a number of mammalian species, as well as in a variety of species that I'll talk about coming up. We ended up taking a variant from mouse because it had been functionally expressed in E. coli and shown to have the highest activity of 4 different variants that had been tested in that particular study. But that was actually the end as far as a good pathway to make myoinositol in mammals. And then the last bit of this was that there had been a paper published that showed that you could have one gene, this INO1 gene, which encodes a myoinositol 1-phosphate synthase from Saccharomyces yeast. That had actually been shown by John Frost at Michigan State to allow you to go from glucose all the way to myoinositol, having just this one clone gene activity. The PTS system brings glucose into E. coli as a phosphorylated glucose 6-phosphate molecule. Then there's an endogenous phosphatase that's very good at dephosphorylating that to give you the product of interest. So instead of having now this natural pathway that was 10 or 12 reaction steps, we only needed 3 recombinant enzymes or recombinant genes in this pathway to give us a compound that could be produced in E. coli. And so it works in the sense that we can make it, but remember our goal is to maximize the productivity of this enzyme. And there are a couple of challenges that you can see, one of which you can see right away, which is that the first intermediate in our pathway is glucose 6-phosphate. And so if I put on my old metabolic engineering hat and say, well, what are the competing pathways for glucose 6-phosphate? Can I just knock those out to eliminate the byproducts? Well, that's essential for growth if I'm providing glucose as my sole carbon source here, which I may want to do for economic purposes. So that's a challenge that we've had to deal with all along, and I'll come back towards the end and give you just a short snippet that's an overview of some recently published work we've done to address that. But in many, many, many studies we've done over about a decade, it's become clear to us that this second enzyme, the myoinositol oxygenase enzyme, is really the enzyme that causes the most problems. And it causes problems for a number of reasons. And I'll try to explain some of those as I go through the talk here. So our challenge was really to figure out, is there a way to actually improve the activity of this enzyme? So I'm going to give you just one highlight of an approach that we took to do this. And the diagram that represents this is shown in this lower right corner here, where the idea was that if we could actually bring this myox protein and the enzyme MIPS or INO1 that encodes, or rather the enzyme that produces or results in the production of the substrate for myox, then we could decrease the diffusion distance between those two and get increased flux through the pathway. And what we actually found is that in using a system like this, we were able to increase the specific activity of the myox protein. And we think that's related to the fact that this is an enzyme whose activity is actually higher when it's expressed in the presence of its substrate versus not being expressed in its presence. And I'm going to come back to that point as well. The main take-home message from this slide, however, is that you can see if we are able to increase the activity of this enzyme, then we actually get higher productivity. So again, this gives us motivation for focusing on that enzyme as the particular bottleneck, or at least the initial bottleneck in our pathway. So I'm not going to talk about many of the other ways that we have attempted to improve the activity of this protein from mouse, but really what we started asking ourselves a few years ago is that actually the best variant of this enzyme to use? We know we want the enzyme activity. We don't have any special feelings towards mouse. I am not a fan of mice personally, so it would be perfectly fine with me if we didn't have anything from mice in this thing to begin with. But the question here is, can we find other variants that have higher activity that could possibly be used in the pathway? So to address this question, we actually made one change to the system, which is that even though we had done a lot of our work in E. coli, we became interested in working in yeast, and so we switched and started looking at yeast as a host for this pathway. And there were a few reasons for this. One is that the compound we're making is an acid, and we know that yeast is more acid tolerant than something like E. coli. So for longer term production, we thought if we can get something that works well in yeast, that will be good. There are even commercially available strains that can be licensed that are particularly acidic, tolerant to low pH, and so there's the possibility from a processing perspective to facilitate the downstream by running the whole thing under acidic conditions. So that was one. The second one is that we were dealing with a system where we actually weren't that surprised that the mouse enzyme was the one that gave us the most trouble. We're taking the mouse protein and putting it into an E. coli, and it's the most distant phylogenetically from as far as the natural sources of the other enzymes. So we thought, well, maybe if we actually have a eukaryotic host, then we'll actually get better expression of this protein of interest. And then we thought, well, this will also give us an opportunity, since we thought that most of the enzymes we'd be interested in would be from higher organisms. This would be a better platform to screen additional variants of the enzymes to see if we could identify something that's more productive. So what I have shown here is the same pathway again, but what I want to point out is that one challenge for us, and one of the reasons why we approached yeast but did it with a little bit of trepidation, is that remember I said to you that the ion-1 enzyme is actually naturally present in yeast. And in this case now there's a specific phosphatase, this INM, that we know is responsible for the dephosphorylation of myoinositol 1-phosphate to give us myoinositol. And so we were concerned about, and you'll see we had reason to be, about whether or not in coming from glucose as a starting substrate, whether or not we could overcome some of the regulatory barriers in terms of how inositol is regulated or maintained within the yeast host. Okay, so again, you'll see as we come back that we had reason to be concerned about that. So we've got this goal now. We want better enzymes. We want more enzymes. And the question then is, well, where do we get them from? How do we find them? Okay, so we know, and everybody in this room knows, since this is a JGI user meeting, that there is no lack of data on sequences. Okay? If anything, I think we got enough. And the problem is, how do we actually get through the stuff that we have? So there's an explosion of data that's coming in, but if you actually compare that with experimental validation of annotated enzymes or a predicted ORFs, an annotated function, functional validation of that function, you can't keep up. And so I had a postdoc from Patsy Babbitt's lab at UC San Francisco who came and brought this perspective to us. And this slide showed this nice exponential increase. Now the scales are off by about 3 orders of magnitude, but at least there was a lot of work being done to functionally characterize enzymes. And then it just sort of stopped. And he's like, oh yeah, there was a grant that ran out at that point, and this consortium couldn't do the work any longer. Right, but this hasn't stopped. We still have more and more and more information that's coming. And there are great tools that actually allow you to do automated annotation to actually predict what the functions are, but the problem is they're not perfect. Okay, so they're pretty good. You can see here, this is showing in the circles here the fraction of the predictions that are misannotated. Again, this is from Patsy's lab. This is reference is cut off here. And the greens are the ones that are done correctly. The reds are the ones that are done incorrectly. And I remember Patsy giving a talk about this maybe about 5 or 6 years ago, and the person who introduced her said, Patsy Babbitt is going to give you the scariest talk you have ever heard about genomics. Okay? And then Patsy got up and basically said, you can believe about half of what's in the databases and about half you can't believe. Now, I personally was quite terrified by this because what we want to be able to do is take these enzymes and put them in the pathways. And if the predictions are wrong, then if I'm making these selections based solely on the predictions, I'm going to be right sometimes, but there's only so much money I'm going to get from federal agencies to keep doing the same thing over and over again. Right, so the challenge that we have is how do we actually navigate this very vast sequence space to find something that is useful? So this was an approach, again, that Michael Hicks brought into the lab from Patsy Babbitt's lab, which is actually around using protein sequence similarity networks. And there have been a couple posters that are up that hopefully some of you have seen who are interested in this approach, where it's not filled with a bunch of information, but it's a mapping approach to allow you to do pairwise comparisons between sets of enzymes if you have a large collection that you want to analyze. And then based on the stringency that you assign for the similarity, you could look at ones that are closer or further apart from each other as far as that similarity is concerned. And the particular advantage of using this kind of approach is that there's a visualization that goes along with it that helps you to map on certain features that can be useful. And I'll give you a couple of examples of those coming up. So Michael actually took this approach and collected at the time what were about 340 sequences in the PFAM database that were annotated as myoinositol oxygenases. We had a mouse one that we knew was actually a functional enzyme, and we had two other variants that we knew from the literature were functional as well. And the mouse one is here in cluster number 7. The other ones are in cluster number 3. And then it was really just a search to say, well, let's do some sampling, one from each of these 7 clusters, the way that they're organized, and just see, are they active or are they not active? Okay, so if we start just at that first level, here's now those 7 clusters, or selections for each of those 7. Cluster number 7 is the mouse one, and we actually did find 2 that in our initial test gave us higher productivities. And I'll point out what's shown here is not glucaric acid, but glucuronic acid. So that's now the direct product from this myoinositol oxygenase enzyme. The other thing that I'll point out is you'll see 2 sets of bars. The white bars here are when we are not providing any myoinositol in the medium. So we have glucose present. We do have a mutant that is supposed to accumulate myoinositol, and so now we're trying to make this product from glucose, versus the gray bars where you see that we are actually supplying about 10 grams per liter of myoinositol into the medium. So that's about 60 millimolar. Okay? And the titers that are shown here are in millimolar. So I mentioned before that we were concerned about the fact that inositol is native to yeast, and so there was a question about whether or not we would really be able to siphon enough material from that node to be able to make this pathway because of the regulation. And you can see in every case how much we are able to make from glucose alone is very, very small. But we can make much, much more from myoinositol, and so that's at least giving us some indication of the activity of those enzymes under idealized conditions, and then we can come back and try to deal with the competition for glucose a little bit later. Okay, so again, so here's our mouse one. The Arabidopsis one, and this is C. neoformans, those were both ones that we knew in the literature should have some activity, but we're actually pleased to see that this totally naive case actually produced an enzyme that was functional. But you can see we actually have 3 others that had no activity or very minimal activity. Okay? So the next question was, could we actually use this small amount of sequence data? We have 7 sequences that are shown here out of 336 possibilities, figure out some key signatures, if you will, and identify something that was better from cluster 2, and could we go back to cluster 3 now with this small amount of information and actually find something that had any activity at all, okay? And I have a colleague at MIT, Tony Sinsky, who likes to say, the best projects are one where you're starting from making 0 and then you make something, because anything divided by 0 is infinitely better than what you started with, okay? So we're looking for infinite improvement here. And then just seeing whether or not we can find some key features that allow us to do better than what we found with cluster 2. So when Michael actually pulled the data together, what he actually did, if I go back a slide, is he took these 3 top performers and said, what's common about them? Because we know that they work well. And so what are the things that seem to be conserved between them, and what are the things that don't seem to matter very much? So on the left-hand side are shown coloring of these different nodes based on the number of residues that are different from the consensus of these 3 proteins within the active site. And the active site in this case is defined as being within 10 angstroms of where myoinositol binds, or this enzyme actually has a di-iron cluster that's essential for catalytic activity. So it's within 10 angstroms of either the substrate binding site or where these 2 irons are. And there are about 25 residues that actually fit that criteria. And so the blue in this case are ones that have 0 changes relative to that. Every time in doing a pairwise comparison of those 25 amino acids, if you find something that's different, it gets a +1 value. And so you can see that, maybe you can't see, I can see here and I can tell you that the yellow says 13. So this guy out here had 13 amino acid residues that were different. It has no activity. Okay, so we're pretty sure that those residues are important. Okay. So what's color-coded here then is a few other samples where the closer you are to the blue or the gray, the fewer changes you have relative to the 3 that we knew were pretty active. And then the other thing that seemed to be important was the length of the N-terminal region. And so this is something we don't fully understand, but it appears in some crystal structures that were available that the N-terminus is a disordered region. But it has to be of a sufficient length, and sufficient is something that we've struggled to define in order for the enzyme to be active. So for these 3 that we had that were active, anything with an N-terminal length, an unstructured N-terminal length, shorter rather than 25 amino acids, actually had no activity. And the best performing ones had these N-terminal lengths that were really around 70 to 90 amino acids. So based on now these 2 features, whether or not you actually have conservation of the active site residues, and then looking at the N-terminal sequence, Michael went back to those 2 clusters, 2 and 3, and picked another enzyme from cluster 2, so something that's closer together. But if you can, maybe on that side you guys can see a little bit better. The original one that we have is a little bit grayer, so it's a little bit more different. And then we went in and picked one that's nice and bright blue, meaning those active site residues are very closely aligned with what the more active variants were to begin with. And again, if we look at the N-terminal length, as you go towards more of the yellow, that's a longer N-terminus. And so we went to here and picked one that was slightly longer than before. And then we picked 3 different variants within cluster 3. Remember, cluster 3, we're starting at 0. So we have less confidence that just picking one is actually going to give us the most desired results. And in this case, we picked 3 where the active site residue conservation was still really, really good. And we weren't entirely sure how important this N-terminal sequence length was. So the 3 that were picked here, one was chosen to intentionally be less than the 25 amino acids we had decided were important. One was chosen to be around 50 amino acids in length. The other one was about 80 amino acids in length. Okay, so we're trying to validate this theory about the N-terminal length being sufficient. So here's now the first 7 samples that I showed you before, and then you can see a new set of data. So in cluster number 2, using those 2 criteria, Michael went in and was able to find another variant that was almost 10-fold, so 8.5-fold higher in terms of its productivity, just based on these 2 features. And then because, again, with cluster 3, we were starting from nothing, he picked these 3. And the one that he chose that had a short N-terminus, again, gave us no activity. And then the one in the middle here had this sort of medium-length N-terminus, and the longer one that seemed to correspond to the length that we found in our more active variants, you can see actually had a productivity that was comparable to what we found with the other variants in the first part of the screen. So if we take those 4 that are most active and then add in the second enzyme, so now we're looking to make glucaric acid, you can see they're pretty comparable to each other. So nothing is really spectacularly better than the Arabidopsis. This was the best one we had identified before, and we actually had done some additional work with the Arabidopsis. But we actually did a pretty good job of picking out something that was active. And in particular, in cluster 3, that's where we started from nothing and then found something that was at least as good as the mouse protein that we started with. So this network gave us the advantage of being able to calculate. It's very fast because we can calculate these pairwise interactions as opposed to doing multiple sequence alignment. So each of those dots and the lines or edges between them is representing a similarity only between 2 sequences at a time. We've got this ability to map on the information of interest, so I tried to show you that a couple of slides ago. And you can have this more interactive nature to either pull things further apart or bring them closer together based on how you set the stringency to decide what properties you think are most important to you. So those are all the good things about it. The bad things about it are that it's just a method. It doesn't actually give us any real information or any insight about where this signal is coming from and how uniform it is across the set because we're doing these pairwise comparisons. It's not a phylogenetic tree, so we have no information about evolutionary history or relatedness in that, for that metric across these. And if you have small changes in proteins, they can have big differences that aren't necessarily reflected in these similarity scores. So I come back to the fact that within this one cluster of these enzymes, cluster 3, that were very close together, we went from having something comparable to the best we've ever seen to something that had zero activity at all. So what we took home from this is the fact that we can use these sort of clustering algorithms, but our goal was to figure out, can we use a small amount of experimental data? So we're a small academic lab, we're not the JGI, right? Can we use a small amount of data to help us to filter through these larger sets of information? So with that as a background, that's actually what led to our JGI synthesis project, and I have the title there. It's long enough that I'm not going to read it, but the goal was to go back now into this network with the information that we have and pick another set of proteins to test and see if we actually could get a higher hit rate this time around compared to our first iteration where we were doing this really blindly just based on where these proteins were located within this network. So I'm going to walk to this side now so the people on this side don't feel so neglected. So the process that we took here is the general strategy here. The first thing was to choose what's known. So we could go into Brenda, the Braunschweiger enzyme database, pick enzymes that have been characterized, as well as ones that structures were available for. So we didn't go completely blind. We started again with 3 in this case that we expected to have really good activity, just to verify that at least the information that was known was consistent. The second thing that was done then was to use what we found previously about these structure-function relationships to establish what we thought were acceptable boundaries. So we know that we want to minimize the differences within the active site, and we also want to try to keep a reasonable N-terminal sequence. And then get close to what is known in this case. Okay, so don't go too far out, but leave yourself the opportunity to be surprised. So this last bit of choosing from the Wild West is to say, stick close to the things that we know, but then as you build these networks, you can see along the bottom, we end up having these clusters that don't seem to be really close to anything. So we picked a few of those just for the purposes of trying to cast a somewhat wider net. So based on that, we actually had 31 candidate enzymes where JGI synthesized for us the genes and coding them. There was a secondary part of this which was about randomizing promoter and terminator regions. I won't have time to talk about that. We had a poster here today. I actually think that data didn't make it in the poster either. But if you're interested in that information, send me an email and let me know, and I'm happy to share with you what we have found so far. And I have Michael Hicks's name on here as well. Lisa Guay is in the audience, and she's here manning the poster, or womaning the poster. How about that? But she's here to talk about the poster and can tell you more about the approaches for, that we use in selecting these enzymes. So this is a lot of data. Of those 29, we actually were able to successfully construct fully functional pathways for, of those 31, we could construct successful pathways for 29 of them. And these are organized in order of descending productivity. So the highest productivity for glucaric acid here, where the bars on the left are when we provide myo-inositol to the medium. So those are, again, consistently higher. And then the darker blue bars on the right are ones where we are not providing myo-inositol, and we're looking for the ability of the cell to be able to compete for that glucose 6-phosphate against the rest of the, or compete in this case for the myo-inositol for those other essential pathways in yeast. So walking through this a little bit, 23 out of our 29 are actually active. So we got about a 70% hit rate here in terms of having active protein that was being produced, meaning that we're actually producing glucaric acid. So we're using as a readout in this case, are we actually making the product that we want? So not bad. So about 70% compared to in the first go round, we actually had 7, 3 of which we knew were supposed to be active, and of the other 4, only 1 was active when we started. Okay, so if you think about it from that perspective, we went from a 25% hit rate in a naive set to about a 70% hit rate in the naive set. We have 2 of these here that are sequences number 20 and 22 that are the 2 that are farthest apart from each other in sequence similarity, but are actually very close to each other in how productive they are. Okay, so again, the point here is that the sequence similarity network gives us some information. It gives us, from our perspective, an organizational framework for thinking about how to interrogate these large amounts of sequence data, but clearly it's not telling us everything, because 2 things that are far apart in the sequence similarity network having the same function means that there are other features that are important in this case as well. We did have 6 of these that we didn't see any productivity for, and the numbers are listed there. The variants, they come from 2 from plants, 1 from a bacteria, 3 from fungi, and I can say we have active variants that are from those 3 kingdoms as well. So it's not that those kingdoms don't have active variants, but these don't. And I'll point out that 5 of those 6, if we look at the PDB entries, are listed as completely uncharacterized. Actually, 2/3 of these are uncharacterized. So most of them are not characterized, and all but 3 actually fall under the unreviewed category in the PDB, meaning they're just based on annotations. Okay? So it's not too surprising. That was actually good that most of the ones that are not active are under this uncharacterized part of this as well. And then the last part is that 9 of our variants actually give something comparable. And comparable, we have the cutoff here at 1.5 grams per liter, and this is over a 3-day fermentation. So 1.5 grams per liter. And our previous best is actually now the second best that we have here. This number 18, the one that's the best, is from mosquito. So not from mouse, not from fungi, not from bacteria, from a completely different source, and has a pretty good activity. So one, of course, big question that you always have when you're faced with a data set that looks like this, especially when you have things that are not active, and for the whole data set really, is whether or not the differences in activity you see, or differences in productivity, are about differences in activity of the enzymes, or are they about differences in expression levels of the enzymes? And especially if you're dealing with naive enzymes, they're being produced in a heterologous host. We don't really know how productive these are or not. I'll point out that our construction method for this was to integrate all of these into the genome of yeast. So the productivity is actually relatively low. When we do this in E. coli, we use high copy plasmids. We get so much protein that a simple SDS page gel tells us everything that we need to know. In this case, we didn't have that benefit. We thought about using Western blots, which are specific, but could we really trust cross-reactivity of an antibody that we can find against 29 different variants of this? We put His tags on these and tried to use anti-His antibodies, and in some cases we lost the activity because the His tags were on the N-terminus end, and oops, I said that was important, and then we put it on the C-terminus end. Turns out that was important too. So what we finally ended up doing was, see, new talk. There's a slide I didn't know was there. Okay, so this is just briefly just to show that the active variants are coming from a wide variety of kingdoms. So across all the kingdoms we tested, we found active variants. The sizes of the circles here represent the titers. Okay, so the bigger circles were the more productive ones. And again, not just were the active variants found across all kingdoms of life, but the more highly active variants were also pretty well distributed. And in fact, the top 3, I told you mosquito was the highest. The next 2 after that, Arabidopsis control, were from a fungal source and a bacterial source. Okay, so this is an enzyme activity that's pretty widely distributed in nature. Okay, so what we decided to do then to figure out what was going on was to do untargeted proteomics. So we actually got a contract with the Donald Danforth Plant Science Center in St. Louis, sent them sample, and they used a completely blind approach to give us just overall protein abundance of the various, the 4 proteins of interest in our pathway. So that was the initial enzyme, the myoinositol 1-phosphate synthase, the phosphatase that cleaves the phosphate, this myoinositol oxygenase, and then the urinate dehydrogenase. And so what's shown on the top here are the first collection are for cells that were relatively high producers. In the middle, we had the ones that were medium producers, and at the end were all 6 of the ones that were non-producers. So this is just a subset for the higher and the medium producer ones, because we are more concerned about understanding why the ones that don't work are faulty, as opposed to, at this point, getting more data about the ones that do work. I'll point out here what's shown on the bottom are the actual titers. These are represented as grams of glucaric acid per gram of myoinositol. And you probably can't see unless you're in the front row, but these numbers are pretty low. They are less than 10%. That's because the data here were taken at 24 hours. And we typically see for this process, the first thing is that the yeast take all the glucose and convert it to ethanol very, very rapidly. And then they will reassimilate that ethanol, and they make most of their glucaric acid late. So this was an early time point because we did want to see what happens before the enzymes, if they were going to be unstable, before they would lose any stability. So the colors that are shown here, the orange along the bottom is the UDH, so that's our bacterial enzyme. That's the last step in the pathway, and then it goes up from there. So the green is the myox, The yellow in this case is the phosphatase enzyme, and then the blue is this INO1, which is the enzyme that's native to yeast. And we didn't knock out the native copy, but we added in another copy. And so what we saw is exactly what we expected to see, which is that there's an abundance of the MIPS enzyme, or this INO1, relative to now our wild type control, which is not overexpressing the pathway. Okay, so I've talked through this enough to give you a chance to see there is not a lot of green on this side. And in fact, of the 6 that were non-productive, 5 of them had no detectable myoinositol oxygenase being produced. Okay? You could be sad about that and say, oh, they weren't expressed, but that actually makes us feel better, because it doesn't mean that there's anything wrong with having chosen them just from the perspective of looking at these sequence characteristics. It means that there's something wrong in terms of being able to express them. So it still could be the case that we're actually able to get active protein. Perhaps we need to change our codon optimization. Maybe we need a soluble fusion partner or something like that to be able to see what the activity was in that case. And then 4 of our 6 non-producers actually have really high levels of the UDH relative to the other enzymes. And that may just be a reflection of the fact that we have a lower overall protein burden if we're not making this myox protein. And so the other proteins are able to be expressed. It also tells us that the other pathway genes did get integrated. So this is very early data. We still have to go back and resequence and make sure that there wasn't a premature stop codon or something like that inserted into the gene. But we do have confidence that the construction method worked, because the other proteins that we expected to see, the recombinant proteins we expected to see, are actually found in that case. So let me summarize this first part, and trust me, the second part is not nearly that long, so don't panic and think you're going to be here for hours. So this sequence similarity network approach actually gave us additional targets that we could use for pathway integration. And I should mention that we also have another enzyme library. So I said that Lisa was in the audience. Lisa's poster talks about the construction of the enzyme library for that first enzyme in the pathway, the myoinositol 1-phosphate synthase. And that screening has been done initially in E. coli. So we did a parallel screening approach of looking at both of those. Now, I mentioned that we're interested in finding better enzymes, and we think that this sequence-guided or bioprospecting approach can be useful for us, but we're not sold on it being the only way for us to do that. We're interested in directed evolution, that is, creating a lot of variations ourselves and then having a screen to be able to find the most active variants for that. And so what we're really hoping is that what this will allow us to do is to identify some of the better starting points for that directed evolution exercise. So even though my colleague likes to say it's better to start with 0, if you're doing directed evolution, not so good to start with 0. You want to start with something and know that you actually can make product, and then see if you can get an advancement from that. So we're continuing to look at these variants, continuing to characterize them, and then our hope is that we'll be able to put them into a directed evolution assay to see, or a directed evolution exercise to see if that will actually give us even better productivity. So in just about the last 10 minutes, I want to talk about another approach we've taken to try to deal with this problem of myoinositol oxygenase. So I've said we've worked on getting every hammer we can find to bang on this myox nail for about 10 years. And we observed something very early on about this enzyme, and I actually alluded to it when I talked about the studies that we had done with the scaffolding. And that's the fact that the enzyme is much more active when it is produced in the presence of its substrate versus when it's produced without its substrate. And when I say much more active, it's about an order of magnitude more active. Okay, so, and this is now in E. coli. If we're looking at this in E. coli, and if we're looking at the mouse version of this myox protein, the other thing that we have found is that the protein is not particularly stable. So we get the highest activity within the first 24 hours. We continue to make product for about 72 hours before it starts to level off. But the enzyme activity is going down and down and down over that time period. And there's a significant drop off over that second 24 time hour period. So those two argue for the fact that we should not necessarily be turning on myox expression at the same time as all the other genes. Okay? And in fact, the plot that's shown on the left is data that's not published. It was in a student's thesis. But this was a student who was investigating this phenomenon. What he decided to do was just to have the first two enzymes in the pathway being expressed from two different inducible promoters. And so what's shown on the bottom is just the time of induction of this myox protein relative to when the other enzymes are being induced. And you can see the productivity here, again, glucuronic acid, so focused on myox, but from glucose, we get much higher productivity if we wait and produce myox later. Now, there's a trade-off. If you wait too late, you actually don't have enough synthetic capacity in the cell. But we'd had this data for a long time. The problem was, who wants to sit around and actually be figuring out when to manually add additional inducers to get now this timed expression? Instead, what you'd like to have is some way of autonomously controlling that induction so that the cell will sense when its myoinositol concentration is built up to a sufficient level, and then it will initiate expression of the myox protein. So I went to a conference in Germany a few years ago. I said, oh, I'd love to have that. And someone in the audience came up to me. This was at Jülich Biotech Center, and Julia Frunsky came up and said, would you like to have a regulator that responds to myoinositol? Would I ever? And she said, we haven't published the paper yet, but we've submitted it for review, and I'm more than happy to share it with you. And they do a lot of work with Corynebacterium glutamicum, and it turns out there's an enzyme, or there's a regulatory protein, IPSA, in Corynebacterium that binds to myoinositol. And in the absence of myoinositol, it also binds to DNA. In the absence of myoinositol, it binds to DNA. And in Corynebacterium, it actually serves as an activator when myoinositol is absent. When myoinositol is present, that binds to the IPSA. It actually then is freed from the DNA. And in Corynebacterium, that turns gene expression off. Well, we actually had a couple of tries of getting this to work and finally figured out if we created a hybrid promoter here where we had binding sites for IPSA, but a consensus -10, -35 sequence for E. coli, we should be able to have something where IPSA is acting as a repressor when myoinositol is absent, and then when myoinositol is present, it actually will dislodge or displace from the DNA, and we will get activation of gene expression. So that would allow us to actually have this myox expression directly controlled by the amount of myoinositol that we have in the medium. So we could test this first by just adding in myoinositol to cultures where we've got now IPSA being expressed. We've got our hybrid promoter, as I described, just making GFP, and we're adding in myoinositol at different concentrations. And what these different lines represent is the strength of the promoter that's producing IPSA. Okay, so again, if that IPSA is going to sit on the DNA, if I have more IPSA, if its promoter is stronger, it should take more myoinositol to dislodge. If I have less IPSA, it should take less. And indeed, what you see is that this is the direction of increasing IPSA promoter strength, where the tighter or the stronger this is, the lower your response is. And then at the bottom, of course, is the negative, no GFP control. And at the top is no IPSA, which is just constitutive expression. So this is great. We see that the sensor actually responds in this dose-dependent manner if I'm providing myoinositol outside the cell. If I'm actually then providing myoinositol inside the cell, so now we provide glucose, and then we have this MIPS enzyme, or again, it's also known as INO1. That allows us to make myoinositol in this case. And again, we see this same trend where when we're able to make myoinositol, we get a GFP response. The lighter bars shown here are the cases where we don't have that gene being present. And you get a higher GFP fluorescence when you have a weaker strength of that promoter being present. So this was good. We now found that the sensor would respond to endogenously produced myoinositol that comes from glucose. So the next step then was to see how does this actually affect the production of our glucaric acid, with the idea that if we're expressing later, we're expecting to see more active myox and hopefully higher titers. So now we've got myox being controlled directly by this regulator, this circuit. And here are again the different promoter strengths. And you can see at the weaker levels, it's not very different from having no IPSA at all. But in the best case, we get actually about a 3-fold increase in the glucaric acid that's being produced. So I've got the reference here. This paper actually was just published, went online with PNAS 2 weeks ago. And one of the more exciting things about it was that we described the construction of this sensor, but then we integrated this with a system that we had previously described and that was published about a year ago now. And that was one where we were back to dealing with this fact that glucose 6-phosphate is this shared intermediate, okay, where we know the cells need it for growth, but we know it is essential for our pathway as well. And again, here are the competing pathways that we can't simply just knock out and say, well, and still allow growth on glucose. And so leaving out a lot of the details here, what we were able to do in this case was to knock out one of the pathways for glucose utilization. So we knock out ZWIF. That's okay. It turns out cells are perfectly viable and happy if we do that. And then if we look at the EMP pathway for glycolysis, we actually created what we called a metabolite valve to dynamically regulate the activity of PFK, first allowing the cells to grow. So that's the necessary growth that I talked about way back on slide 2. And then when we actually have built up enough biomass to be able to down-regulate the expression here, that should allow us to build up glucose 6-phosphate and have more of that available to go into our product of interest. And so just one slide from that paper, what we were able to show is that under conditions where we have actually usually the highest production of glucaric acid for our smaller scale, we actually are able to use this system. And in this case, we're triggering that valve by using a quorum sensing system. So it's completely autonomous in this case. And the timing is actually dictated by how strongly expressed the synthase is for the signaling molecule. This is the ESA quorum sensing system. We actually got this from Cynthia Collins, who's also at the meeting here. And what's shown here is the strength of ESA-I. So weaker expression means later switching. And you can see in our best case, we actually get, it's at the bottom. The green is glucaric acid. So the best case, we get only glucaric acid and no acetate. These are our controls. So our wild type control actually makes only acetate and no glucaric acid at all. So it turns out if you actually combine both of these strategies, and individually they increase productivity from 2 to 3-fold, but the best case now, if we combine them together, we get a 10-fold increase compared to our negative controls where we have neither the PFK valve being triggered nor the MIOX control, or the MI-controlled expression of MIOX relative to that. And this 2 grams per liter in a bioreactor are the highest titers that we've seen in this particular strain of E. coli. So great. Susanna is stepping up. This is my last slide. So summary and next steps. So the first part of this is that I started with this bioprospecting approach, and it does allow us to identify enzymes that can give us pathways that don't exist in nature. But we really need tools to help us filter through the massive amounts of sequence data to find the best candidates. And I have best in quotation marks here because it's not always clear to us what it means to be best. We know we want things that are most productive, but there are other properties of these cells, of these enzymes that matter as well. I told you this last bit about now being able to use dynamic control as an alternative way to maximize titer and yield. And we have 2 different ways of doing that. But really, the point of showing you that data was to come back to where we want to go with this system of MIOX enzymes. So the first thing that we have to do is more characterization. All the data that I showed you with those MIOX proteins, that set of 29 that we got from JGI, that was all screening at very small scale in a biolector. So for those of you who are familiar, it's a 1 milliliter reactor volume. We don't know how these are going to behave as we move up to shake flask scale or even a bioreactor scale. So we want to do that, but we also want to look at them under different conditions. So we actually don't know if any of these other enzymes have that same feature of the mouse one, where they're more active when their substrate is present versus not. Ideally, we would find something that is not so highly dependent on the substrate concentration for its activity, and perhaps that's found within this set of enzymes. And so we're really excited to test now all the different conditions where we know the mouse one has given us wildly different, wide and wildly different activities, and see if that's a trend that holds for the family at large, or if that's a unique feature of the mouse proteins. We actually still have support from JGI. We have capacity for more synthesis, and so we want to actually synthesize more genes or more candidates, but we want actually to figure out what are the best things for us to synthesize. So again, going back and figure out for the ones that didn't work, what is it about them that made them not work, and seeing if that could be information that we put back into how we interrogate the network. And then as I mentioned before, what we're really interested in doing is using this to allow us to find the best starting points to see if we can get something even better using directed evolution. So I'll just briefly acknowledge all the folks who did the work. Lisa Anderson was largely responsible for all the JGI screening, and that was continuing the work that Michael Hicks started. Lisa, I mentioned a couple times, is in the audience and has done the library on the MIPS protein. And then Stephanie and Apoorv were the ones who did the work I described at the very end about dynamic control. And then lastly, again, the synthesis work was through a JGI synthesis grant, and then the other support has come from the NIH and from NSF. And with that, it looks like I have 10 minutes or so to take questions.